All right, class, it's time to study biology. Been missing you guys, but before we start, I do want to finish my lunch. So just give me a second here and we'll finish. Here we go. Want good digestion, so. Nothing quite like good digestion. Of course, I was able to do that. Well, it's because of cell communication. Okay? So that's what we're looking at today, cell communication, specifically synapses, but we'll get a general overview too. All right, so when you're born, you're one cell, and you guys know every cell has all the genetic material to make as every other cell. So what happens? You have to differentiate, okay, and different cells become different things. So there's got to be external signals from the environment, okay, or from nearby cells to tell that cell what to do, what to produce, what to turn on, what to turn off. All right, so cell communication is key. All right, that's differentiation. All right, now, external signals, okay. How can cells communicate? Well, first of all, there's direct contact. Okay, so some cells have gap junctions, and animal cells, which are little openings between the cytoplasm. So small materials like ions and small molecules can just go right from one cell to another. Okay, plants have what's called plasmodesmata, which are channels from cytoplasm to cytoplasm. So that's a kind of a direct contact type of communication. Okay, we're going to look into synaptic today, which is like nerve transmission or neuromuscular transmissions, okay, using neurotransmitters. Okay, and then next week we'll talk about paracrine and endocrine. Paracrine is when some sort of chemical is released and it affects the cells that are nearby to it. Endocrine produce hormones. You guys know what to make a hormone. Okay. Don't pay her. All right, you guys, I already told you that joke, I think. The endocrines, okay, they secrete chemical messengers which can travel through the blood and get to tissues and turn them on. All right, so the basic overview of all this cell communication is this. Okay, you get an external signal. Now, it's sort of so they give that term a ligand. Okay, so that's the term that you should just kind of remember. I'm not sure where it came from. That's going to signal into a receptor, usually a transmembrane protein, okay? Or if the ligand is lipid soluble, okay, then it can go through the membrane and be an intracellular type of delivery, okay? But most cells have this transmembrane protein which has a receptor which the ligand will fit into, okay? Then that receptor usually has some sort of transformational change in its structure, which will key secondary messengers, and then the secondary messengers will then have elicit a cell response. All right? So specifically, I looked at this today. I thought this would be a great opportunity to show a neuro transmission, okay? Because it kind of fits some of the cell communication, and also we can review some other concepts too. All right? So here's what I got. Here's a nerve cell. Here's an axon of a nerve cell. Okay, so here's a whole nerve cell. So nerve cells consist of a dendrites, which pick up the signals, like a cell body, okay, which of course would have the nucleus inside of it. I'll go right here. And then they have an axon, which the impulse will travel along, and then it hits the next neuron, okay, and it triggers it. All right. So here's what happens. Okay, so we got say a nerve or a voltage transmission going down here, okay, being elicited this way. Okay. At the end, okay, this is called a voltage-gated ion channel. Okay, when this voltage gets to the end, okay, it triggers a conformational change in receptor molecules. Okay, an ion gate which allows calcium into, okay, the end of the axon. Okay, so calcium comes rushing in. Okay, when that happens, the calcium reacts with these vesicles, which are filled with some sort of neurotransmitter. Now, a real common one is ACH or acetylcholine. Okay, so it triggers this acetylcholine vesicles. Okay, it causes them to tether to the end of the membrane, and you guys remember the term exocytosis. Okay, so it's going to connect, and then it'll dump out this acetylcholine into this space. It's called the synapse, synaptic space. It's an actual, these neurons do not actually touch each other, just that small little space in there. Okay, and the acetylcholine gets into this space then. Okay, and then it is the ligand, or the signal molecule, okay, which fits into some sort of receptor. Okay, it's a receptor, usually a transmembrane protein, okay, causing a conformational change. Now, with this ligand-gated receptor, okay, I'll take a little spot here and let's blow it up here. Of course, you notice here a phospholipid bilayer. Okay, so lipid head here. Hydrophilic here, of course, hydrophilic here. So this is, say, outside the cell, inside the cell. Okay. And then these hydrophobic tails. Okay, so of course, because of that, you can't have ions can't get through that hydrophobic layer. Big molecules can't get through there. Anything that's polar can't get through there. Okay, so those bigger molecules have to get through with either facilitated fusion or active transport, or some sort of mechanism to get across. Okay, they can't just diffuse freely. Okay, so that brings us to a nerve cell in itself. Now, a nerve cell itself has a polarity to it. Okay, so if you look here, I just put it in this area. It has an active transport mechanism here. Okay, which is called the sodium-potassium pump. I know we've talked about it before. It's a pump using membranes again to pump 3 sodiums out and 2 potassiums in. It's active transport, so it goes against the gradient, okay, and it uses, of course, adenosine triphosphate. So it's energy, cost energy, okay, and it goes against the gradient. But what it does is it pumps these sodiums out, 3 sodiums out for every 2 potassium in, so you get a difference in charge. So you end up with actually a potential negative 70 millivolts. Okay, so it's negative on the inside, positive on the outside. It does cost a lot of energy to do this. It's rumored it's about 1/3 of the energy you use is to keep this up in throughout your body. Okay, but it sets this up, okay, for a nerve transmission. So what will happen is acetylcholine will get in here. Okay, so ACH comes in and it binds into that transmembrane protein. And when it does that, it causes a conformational change which opens the gates. Okay? When that gate's open, of course, then you have more sodium on the outside than the inside, so the sodium rushes in. Okay? And of course, when it rushes in, it causes what is called a depolarization. So actually when this happens, it goes from being negative 70 millivolts to a positive 30 millivolts. Very quickly. Okay? Okay, so you get this depolarization, okay, and that does, it causes triggers this throughout the whole membrane. So what you get is a wave of depolarization which moves down the dendrites, and then it moves down, where these sodiums are all rushing in, okay, and it goes down and it goes all the way to the end. Okay, now it comes back here. We started with that down here. So you get this wave of depolarization, and then you get the same thing happening here. Okay? Same thing happening here as which went on here. So you get another, okay, dumping of this ACH and triggering the next neuron to fire. Okay? Just a little specific on nerve cells. Nerve cells have some other complexities to them. They have these called Schwann cells that go on the outside of them, okay, which means they're kind of fatty cells. What happens is you get kind of a jumping. That's called saltatory conduction, okay, which means it travels a little bit faster. Okay, that's just a little sidelight. Now, ACH, okay, there's enzymes here which break it down pretty quickly. Okay, so once it causes that to fire, those ACHs are brought away. They can get recycled back in. Okay, but their action is short term. All right. So what you can have here, this would be called